और आज हम एक बहुत ही इंपॉर्टेंट टॉपिक पे बात करने वाले हैं जो कि है माइटोसिस में नंबर ऑफ क्रोमोसोम्स इन डिफरेंट स्टेजेस। अब ये माइटोसिस और मियोसिस देखिए अपने आप में ही बहुत इंपॉर्टेंट है, सेल डिवीजन। एवरी टाइम यू आर गेटिंग क्वेश्चंस फ्रॉम सेल डिवीजन। लेकिन जो कन्फ्यूजन रहती है वो हमेशा रहती है कि नंबर ऑफ क्रोमोसोम्स कितने हैं इन डिफरेंट स्टेजेस ऑफ माइटोसिस। तो देखिए जब हम माइटोसिस की बात करते हैं, माइटोसिस वी नो कैन बी डिवाइडेड इनटू टू ब्रॉड फेजेस। सो ये देखिए, व्हेन वी टॉक अबाउट द माइटोसिस, we know mitosis itself can be divided into two different phases. One is called as the interphase, and one is called as the M phase. And we know that interphase can again be divided into different subphases, and similarly the M phase can also be divided into different subphases. So look at this, when I talk about the interphase can again be subsequently subdivided into, we have the G1, we have the S phase, we have the G2 phase. We know G1 stands for the gap 1, we know S stands for the synthetic phase where DNA synthesis takes place, and G2 we know it stands for the gap 2. And we know in the S phase DNA replication, G1 there's a lot of growth, G2 maybe also there's a lot of growth, there's a lot of protein synthesis which is taking place. All these things are very, very clear. When we talk about the M phase, the M phase we know can again be subdivided into 4 different subphases. For example, we have the prophase over here. Similarly, we have the metaphase over here. Similarly, we have the anaphase over here, and we have the telophase over here. And by the end of the telophase, we know the karyokinesis is followed by cytokinesis. So that means by the end of the telophase, we get two daughter cells. So this is the situation that we have. Now any cell which has to enter mitosis, we know it will enter mitosis from the G1 side. That means a cell will enter mitosis over here. It will enter the mitosis in the G1 stage. Now if I give you the example of a human somatic cell, अगर हम अपनी body cell की बात करें, हमें पता है जो हमारी body cell है, our body cell is a somatic cell and it is a diploid cell. That means in our body cell we have 2n is equal to 46. Look at this, we have 2n is equal to 46. This is the situation over here. We have the 2n is equal to 46. That means in our somatic cell, ये जो हमारी body की cells हैं, यहाँ पे we have 23 chromosomes which have been contributed by the sperm. 23 chromosomes have been contributed by the egg. They fertilize, we get a zygote. The zygote divides, we get an embryo, and the embryo gives rise to the entire organism. That means 23 of sperm, 23 of egg, and now we have a somatic cell which is a diploid cell with 46 chromosomes. So let us not beat around the bush. The situation is very clear. We are starting with a human cell which is a diploid cell and which has 46 chromosomes. 23 are coming from the mother, 23 are coming from the father. So situation is very clear. Now if this cell has to divide, it will always enter what is the G1 phase. It will always enter the G1 phase. That means any cell which enters mitosis will come over here. That means in this cell again we have 46 chromosomes only. This cell will also have 46 chromosomes. And now after the G1 phase, the cell will enter the S phase. S phase में we know this is characterized by the DNA replication. तो यहाँ पे क्या हो रहा है? यहाँ पे हमारा DNA replication होगा. The DNA replication will take place over here, and after the DNA replication, most of the students, शायद मैं भी अब जब स्कूल में पढ़ता था, when I was at your stage, I used to think that after the S phase, the number of chromosomes will double. क्योंकि each chromosome is being replicated, so it looks natural that the chromosomes after the S phase will become 92. Is it so? क्या आपको लगता है कि number of chromosomes will become 92? और देखिए ऐसे क्वेश्चन आते हैं हमारे नीट में. The same question has been asked in the NEET examination. Look at this situation now. I'll take a new page. That means in the S phase, if you look at the S phase over here, we know in the S phase after the DNA replication, what happens? So any cell which enters the S phase, initially it has 46 chromosomes. And after the S phase, the DNA is replicated here. The DNA replication will take place. And after the DNA replication, what do you think? That the number of chromosomes will become 42. This is not the situation. This is a wrong answer. We know 92 is a wrong answer, because you'll be surprised now. You'll be surprised. शायद आप आश्चर्य रहोगे कि number of chromosomes will still remain 46 only. That means even after DNA replication, the number of chromosomes will still remain 46. How this is possible? How this is possible? Yes, this is possible because if you look at the situation over here, if I say in the S phase, if DNA replication is taking place, see, this is a chromosome and this is replicated now. We know this is replicated. Chromosome number 1 is being replicated. Similarly, we have chromosome number 2. This chromosome number 2 is also replicated. Similarly, we have chromosome number 3. The chromosome number 3 is also replicated. So look at this, chromosome number 2, chromosome number 3, and similarly, we will have up to how many? We will have up to 46 chromosomes. That means all the 46 chromosomes have replicated. So this is the chromosome number 46. So 1, 2, 3, 4, up to 46. Each chromosome has replicated. What do you mean? When I say chromosomal replication, that means it is DNA which is being replicated. It is being DNA which is being replicated. So that means each chromosome now has 2 DNA molecules. It has 2 DNA molecules, but they are not free. These 2 DNA molecules are not free. The 2 DNA molecules are attached at the centromere. You can see over here, these 2 DNA molecules after replication are still attached at the centromere. Look at this. They are still attached at the centromere. That means, what do we call them as? So can I call here that each chromosome is what? Are they 2 chromosomes? So do you think now each chromosome has become 2? No, it is not 2. The chromosome still remains 1. That means even after the S phase, even after the DNA replication, this total structure still is a single chromosome. This is still a single chromosome. But the single chromosome has 2 sister chromatids. What we say now, that this chromosome now has 2 sister chromatids. So this is the sister chromatid number 1 and this is the sister chromatid number 2. So each chromosome has 2 chromatids, or it has 2 sister chromatids. Similarly, that means after the S phase, we still have 46 chromosomes, but in terms of number of chromatids, they will become 92. So how many number of chromatids do we have? The number of chromatids will become 92. So this is the number of chromatids, which is 92, and this is the number of chromosomes, which still remain what is 46. So the picture should be very, very clear. There should be no confusion. And as a thumb rule, let me clarify this, that we know the number of chromosomes is always equal to the number of free centromeres. The number of chromosomes is always equal to the number of free centromeres. So the situation is very clear. That means after DNA replication, we still have 46 chromosomes. You can see here, because the number of centromeres are still 46. And each centromere is attached to 2 sister chromatids. So total number of chromosomes still remain what is 46 after the S phase, but the number of chromatids will double. They will increase to what is 92, and each chromosome has 2 sister chromatids. So let us not beat around the bush. Let us move forward now. Let us move forward. So we know the situation over here. That means after the S phase, what is the condition now? Now you know this. After the S phase, when the DNA replication is complete, the number of chromosomes will still remain 46, but the number of sister chromatids will double to what is 92. The number of sister chromatids will double to 92. And now the same number of chromosome will enter the G2 phase. The same number of chromosomes now will enter the G2 phase. Look at the situation. When I start with the G2 phase, which is the gap 2 phase, look at this. So in the G2 phase, if we talk about the total number of chromosomes, they will still remain what is 46, but the number of sister chromatids will double to what is 92. So situation is same as at the end of S phase. Now after the G2 phase, now the cell enters the prophase. Now the cell enters the prophase over here. This is the prophase. The prophase is part of the M phase, as we have seen. The M phase is divided into prophase, metaphase, anaphase, and telophase. So we are talking about the prophase. The cell enters the prophase. Still, the number of chromosomes will still remain what is 46, and the number of sister chromatids will still remain what is 92. They'll still remain what is 92. So same situation, you talk about the G2 phase, you talk about the prophase, the number of chromosomes will not change. The number of chromosomes from G1 to S phase to G2 phase to prophase, they still remain the same. That is 46 because the number of centromeres are still 46. After the prophase, we know the cell enters the metaphase. So look at this, we take a new page, the cell enters the metaphase. And we know in the metaphase, all the chromosomes will align at the equatorial plate. So we have 46 chromosomes. And we know each chromosome is made up of 2 sister chromatids. तो कैसे बनाएंगे इसको? ये देखिए, ये हमारा equatorial plate है. So each chromosome now will align at the equatorial plate. This is the chromosome number 1. This is the chromosome number 2. This is the chromosome number 3 we have. This is the chromosome number 3. Similarly, up to 46 chromosomes. This is the chromosome number 46. So you can see here the number of chromosomes in the metaphase will still remain what is 46 only. But the only difference is that each chromosome is made up of 2 sister chromatids and they are still attached at the centromere. They are still attached at the centromere, and that is why, because the number of centromeres is 46, so the total number of chromosomes also remain what is 46, and that is the thumb rule that we know. And we know in the metaphase, ये जो centromere है, this is attached to the spindle fibers. We know we have the spindle fibers which are coming from the opposite poles over here. We have the spindle fibers which are coming from the opposite poles. They are attached to the centromeres over here. Look at this. So this is the condition that we have in the telophase. Still 46 chromosomes, and at the starting also we had 46 chromosomes. Replication has taken place, but the chromosome number does not change, still remains 46. This should be very clear. Examiner can confuse you. So no confusion over here. And now after the metaphase, the cell enters the next phase, which is the anaphase. Now this is important over here. Anaphase, and we know in the anaphase what is happening. The sister chromatids have separated. So look at the situation. If I draw it over here, again, look at the situation over here. Look at these chromosomes. We know these chromosomes now. Look at this. This is the condition that we have. This is the 46 number. Remember 46 number. This is the number 1 that we have. So do we still call them as sister chromatids? No, we will not call them as sister chromatids. In the anaphase, the sister chromatids have already separated and now they become individual chromosomes because the number of centromeres have now doubled. This is independent centromere, this is independent. Look at this situation over here. So when we talk about the anaphase, in the anaphase, as you can see, 46 chromosomes will move to one pole. These 46 will move to one pole and the other 46 will move to the opposite pole over here. So what is the total number of chromosomes that we have? The total number of chromosomes in the anaphase now become 40, that is 92. The total number of chromosomes in the anaphase will now become 92, and we will not use this term sister chromatids now. Hereafter we will not use the term sister chromatids, we will only use the term what is the chromosomes. So in the anaphase, from 46 in the metaphase, now we have 92 chromosomes and they are still in the same cell. And after the anaphase, we have the next phase, which is the telophase. We have the telophase, and in the telophase, as we all know, these chromosomes, 46 chromosomes will move to one pole, the other 46 will move to the other pole. If I draw this diagram over here, if this is a cell, we know these 46 chromosomes, they have come to one pole, the other 46 chromosomes have moved to the other pole, and we know by the end of the telophase, the nuclear membrane reforms. The nuclear membrane reforms around these chromosomes. Look at this. The nuclear membrane has reformed, and we know now at the end of the telophase, the karyokinesis is complete, the nuclear division is complete. But all the chromosomes, even though we have two nuclei in the same cell, that means the cell still contains 46 plus 46 chromosomes. So that means that if you talk about telophase, in the telophase also, we still have how many chromosomes in total? We still have 92 chromosomes. We still have what is the 92 chromosomes. They are all present in the same cell. Cytokinesis has not taken place as yet. Two nuclei are present in the same cell. So by the end of the telophase also, the cell still has what is 92 chromosomes, and now by the end of the telophase, the karyokinesis is followed by cytokinesis. That means now the cytoplasmic division will also start. We know there's a cleavage which forms in the center. This cleavage or penetration deepens, and finally the two ends will meet. And we get two daughter cells. We get two daughter cells over here. Look at this, with independent nuclei, and each nuclei has 46 chromosomes. 46 chromosomes. So from a mother cell which had 46 chromosomes, we are getting two daughter cells which again each has 46 chromosomes. So this should be very, very clear. That means if you look at if I sum it up from starting of the G1 where we had 46, DNA replicated in the S phase, we still have 46. In the G2 phase, we still have 46. Prophase, metaphase, still 46. But when we come to the anaphase, what happens? The sister chromatids have separated, 46 to one pole, 46 to other pole. The total number of chromosomes increased to 92. The same follows in the telophase, and by the end of the telophase, we know when cytokinesis takes place, we get two daughter cells, and the number again resumes to what is 46. So I hope you have understood how chromosomes number would vary in different stages of mitosis. But before we end up this lecture, there's one more clarity that you have to have. We know mitosis is called as an equatorial division. Now this is also important. You have to know why mitosis. Why this mitosis is called as a equatorial division. Whereas if we talk about meiosis, we know meiosis is called as a reductional division. We know this is called as a reductional division. Now why this is so? Now when we talk about mitosis, we know in the mitosis we always start with 46. The mother cell has 46, and at the end we are getting two daughter cells. Look at this, we are getting two daughter cells and they still contain what is 46. That means if you look at the number of chromosomes, the number of chromosomes are equal in the mother cell as well as the two daughter cells, and that is why, because the number of chromosomes are equal, that is why we call it equatorial, equal number of chromosomes in mother as well as the daughter cells. Whereas if you talk about meiosis, in the meiosis 1, which is the reductional division, the number of chromosomes will half to what is 23. That means if we talk about the meiosis, in the meiosis we start with 46, and by end of the meiosis 1, we get two cells and each has what is 23, and that is why this is called as what is a reductional division, because the number of chromosomes have reduced to half.